Hello, good evening, everyone, and welcome to the third webinar in our series with Dr. Gustavo, all about genetics and regeneration. Thank you for bearing with us. I know we postponed last week's webinar due to the football, so everyone could switch off and watch that. So we're just pushing all the webinars back by a week, so the final one will be first week of August. But for this evening, we are going to be discussing epigenetic regulation and gene expression in aging. No, sorry, I've got that one wrong. Tonight, we're discussing key biological pathways driving age and regeneration. So I'm now going to hand over to Gustavo to get us started. If you do have any questions, please pop them in the Q&A below. And let's get going. Thank you ever so much.
Thank you, everyone, and thank you, Chloe, for the introduction. Also, for making possible for us to be together in this theme. I will start sharing my presentation here, but again, just as Chloe has just, please do not worry if you need to do any questions or to forward any questions in the meanwhile. While we're presenting, we can do this interactively. Obviously, there will be time in the end, but if you need to ask anything, just please do so. It's always a pleasure. Well, so now we're in the middle or exact middle of uh, our series on genetics, and this is the opportunity for us to go from the basic genetics we have started to see, and move towards the clinical practice. So the next two webinars are going to be more into the clinical utility of genetics and the clinical utility of genetics in regenerating and longevity. But today we're going to make this bridge. We're going to close the bridge between them two a bit further. I know we have spoken about some clinical aspects and clinical utilities or connections to genetics, but now there are some things which are essentially um, real life alterations that patients face throughout their lives, and how these biological pathways and biological mechanisms drive aging and regeneration. So again, genetics is a hard thing to do or to discuss, and also I know that sometimes genetics is not as clear as it should be, or as clear as we'd like to be. But I would like to then take this opportunity to go a bit deeper um, in genetics. So this is the whole aim of this webinar. And therefore, I'm going to use a bit of time to, while we are in this third webinar and we have these two other ones to go back a bit in um, and summarize what we have seen in the previous ones. And mainly because essentially we have covered DNA and RNA very completely, thoroughly in the two previous webinars. And I really think that it has to come along with us each time we go back to the practice. It is important for us to go a bit backwards when we go forwards with the practice. We go backwards a bit with the genetic implications or the basic implications. Sorry for the long one. These are the two main references for today. So the Hallmarks of Aging of 2023, and one example of application, which is the PRP you all know. But we're going to focus very much on the Hallmarks of Aging. This is a very important seminal paper, so you all need to read if you haven't. And without further ado, let's go to the basis review. So we are reviewing then the fact that all the regenerations and all the regenerative techniques, they do rely on the mechanisms which are involved in essentially everything related to DNA replication, DNA maintenance or proofreading, and the overall stability of the DNA, and DNA expression into RNA, and therefore the alterations in the function of the cells. So let's start with the DNA replication. DNA replication happens depending on, firstly, a signal from outside or inside the cell that prompts DNA to separate. And then we have this whole mechanism happening in which the strands are separate, and we have the DNA polymerase passing through the DNA, replicating it. In the meantime, we have both the DNA polymerase 1 and 2, but mostly the DNA polymerase 1 going in the opposite direction, doing corrections to the DNA. So this is one thing that obviously has to do with the regeneration. This means that each time a cell divides, we need DNA to be replicated, and we need that replication to be controlled, both in terms of happening, but also in terms of keeping DNA stable. That takes us to the cell cycle control. So the cell cycle controls it. Control means that essentially regeneration is not cells dividing uncontrolled, it's controlled renew. So regeneration always happens when we have cells dividing at a controlled pace and responding to the correct stimulus. Uh, DNA per se is a molecule that stores information, and this is essentially what we have as part of the function of DNA in the cell and in the overall functioning of the biological processes. But DNA also serves as a molecule that promotes hydration. So we have DNA as a biological entity keeping our cells working and keeping information. But when we apply the PDRNs and polynucleotides of sorts clinically, like in the practice, the main function it has is hydration, although we do have some other things we have discussed previously, but hydration is one central thing of DNA utility, especially because it simply attracts water. It keeps water along the backbone of the DNA. And then moving from DNA to RNA, what we have is that not all cells are the same, although they do have the same DNA, and this happens because DNA expression or the RNA production happens differently in all the cells. That also means that we have a mechanism in which the RNA polymerase now is the one that binds to a region in the DNA called promoter, and this promoter region is there for each one of the genes we have. It interacts, so the RNA polymerase interacts with other regions of the DNA and other molecules, and the main ones are the transcription factors. And all of that is involved in controlling whether a certain region of the DNA produces or not RNA. And this is what we discussed also about epigenetics, because epigenetics also takes a place in controlling how well and how much function the RNA polymerase has when it binds to the DNA. So that is all the revision we needed to do so that we can go into finally the hallmarks of aging. So aging, and now we're leaving the DNA behind just to focus on aging as a whole. Aging is a process that happens across time, and it's kind of seen as a process that has markers because we count it in years or we count it in days, so it's seen as a as a numbered process. But it's also important for us to see that there's overlap across the times we are aging, across the time we're aging, and also this is a spectrum. And what I mean by a spectrum is that we have different systems in our bodies and in our cells aging at different paces due to different exposures, due to genetic predisposition, due to different processes happening inside our cells. And one of the most important, perhaps, thing or perhaps summary of all that is the fact that we see the outside or we see what is above the clinical horizon. And um, we see several ways of aging. We see several markers of aging which are physical. We see the clinical part, so we see above the clinical horizon. So we might see the skin being wrinkled. We might see muscle atrophy, muscle loss, hair loss, cognitive decline, and so on and so forth. But obviously, all that happens because we have some systems below the clinical horizon, which are progressively changing and progressively aging, so as to say. Um, And this is the interest of understanding genetics, understanding molecular biology or molecular genetics, is actually to know which and how these systems are altering below the clinical horizon and how they will impact in the patient's overall well-being and also their perception of aging, and also obviously our perception of aging when we have a patient that needs treatment. They might need treatment for their cognitive decline, but also might need treatment for the skin wrinkling. So obviously this also goes to the perception of the very patient on what they need to cope with the aging and what they need to promote longevity overall. And then finally, we have the hallmarks of aging. So the whole paper I've mentioned is, as I've said, a very important paper when it comes to understanding longevity, because it's called the hallmarks of aging, but it is indeed the hallmarks of longevity as well, because we are trying to counteract many of what or much of what happens during aging, or at least manage it, as it's more appropriate to say. And what do I mean by that? So, as I've said, so the aging process, it's going to be a result from the progress of accumulation of molecular and cellular damage. So, all you see around this are going to be foci of damage that can happen and lead to aging. Obviously, this happens in the normal human being, but as I said, um, the aging process is a continuum. So it's not we're not dealing with quantitized things, and this is majorly because one of these processes might be happening faster in one individual and slower in the other one, and in another person it changes the aspect. And that means that some people might be experiencing different types of aging and different different progresses in aging as a whole. So one of the main things that we need to take into consideration is that we're not dealing, as I've said, with one thing happening, but all these things happening at different paces. But obviously they are connected. So all the mechanisms you see there, they are connected. And they all lead to one end, which is essentially how the cells lose their function and how the body overall decreases function. And this is essentially the gradual loss of function the body has across time that leads then to the aging as it is perceived. I'm going to um, focus here, not in the whole presentation, but focus now in the aspects of this aging process, which are the primary ones, but also things that we have already discussed or are involved in genetics. So as for genome stability, telomere attrition, and epigenetic alterations, what we see here are things that are involved or are related to how DNA behaves. So as we lose telomere, so telomere attrition, which we're going to discuss later, this leads us to less genomic stability. And as you have seen in the previous slides, when we have in our DNA essentially um, part of this DNA being expressed in the form of a certain RNA molecule that then becomes a protein, we need this whole chunk of DNA to be intact. So as we lose genomic instability, we go on losing functions in the cell. And this is to some extent due to also losing the capacity to express some proteins. Not many or not all, but some specific genes and some proteins cannot be expressed. And we're going to see this distinction later, which is essentially seeing how the alterations in gene expression, which is one concept we have seen, it's both a reason but also a measure of how individuals have aged. And obviously, as we lose telomere, we lose genomic instability, we lose genomic stability. As we have our epigenetic alterations, we also lose function of some cells. And that all might lead to the incapacity, or the primary thing might lead to the incapacity of some cells to proliferate. So as you can see here, we have some primary reasons, which are DNA maintenance and then also obviously RNA expression from that DNA, and they lead to both the antagonistic side and also the integrative side of the longevity and the hallmarks of aging. So things that are inside our DNA and the progress of aging inside each one of the cells we have, they're going to lead essentially to the deterioration of how the body functions as a whole. And this is what is perceived as aging, and this is what is perceived as well as a focus and a aim in longevity overall. As for the practice of how this happens, what essentially happens is that across time we have normal DNA damage. So DNA damage is something that happens normally, and it might happen from different sources. However, what is very important to understand is that when DNA damage happens, we have many fates for a cell that has been damaged. The main or the normal fate for a cell to happen, or for a cell that has been damaged or has had its DNA damaged, is that the cell stops to divide. And this is the cellular senescence we have discussed in the previous slide. This leads per se to less cells being possible to be produced. So we have less tissue being produced, being regenerated in that patient. So as you can see here, alone this mechanism or the cellular context and consequences from the loss of DNA stability or the production of a certain protein from that DNA stability, that per se, so if we have DNA damage, that per se will lead eventually to DNA not being produced in that cell, not expressing a certain protein, and that leads to cells being lost. So these cells, they are lost because they do no longer proliferate, and this was one of the concepts we have seen. But also, obviously, once these cells die in a different way, that also might lead to alterations in signaling, in inflammation or leading to inflammation, and also deregulation of how the cell functions while the cells are alive. And these two things obviously are going to interact. What is more important about this is that as you see, when we have DNA damage, this is something very inside the cell, but this has consequences on the outside and the overall functioning of the body as a whole. Well, and this takes us to the separate concept of cellular senescence, which is the concept essentially of how cellular senescence happens. So essentially, whenever we have any stress in the cell, it might be a direct DNA damage, it might be DNA damage due to telomere loss, or it might even be alterations in the function of the cell, let's say a oncogene activation, an alteration in the mitochondrial function, and so on and so forth. All these things, they normally should lead to cell arrest. So we have mechanisms in the cells that they lead the cells to arrest, like the P53, which is very widely known. They should lead the cells to arrest. And this is the main center of us losing the capacity to repopulate and remake the function of tissues, which is we have DNA damage, we have cellular damage, cells stop dividing. This is the concept of cellular senescence that leads to alterations in longevity and leads to alterations in the appearance of aging. So this happens to the skin cells. This happens also to the brain. This happens also to the to the epithelium and so on and so forth. So less cells dividing. Obviously, this is a normal mechanism, but less cells dividing means less cells to replenish the tissues, but also means that the tissues start to do different types of proteins or different levels of certain proteins. So this also alters how the tissues function as a whole, and not only simply the fact that we have less cells proliferating in these tissues. Second, important correlation to the hallmarks of aging is the inflammation. So we have a dichotomy, and this was a question we discussed in the end of the previous week or the previous webinar week, which was, well, the whole concept around many of the longevity processes or many of the aesthetics process is to induce inflammation. And how that differs or how that can be a problem or how can that be a solution when it comes to essentially um, how the good results can be achieved. And we have discussed essentially that while inflammation might happen and it might lead to cell proliferation, it might lead to tissue reorganization, reordering through the normal process of the cell balance. However, as we can see here, we have a concept that has emerged in the later years, which is the process or the concept of inflammaging, which is essentially the inflammatory process, which is persistent and low-grade inflammation, but also that interacts with acute inflammation that is called inflammaging. So kind of a nice name to put into the processes that come from the fact that inflammation also interact with the regeneration overall and also the stress of the organism. So as you can see, things as visceral obesity, the very fat tissue that produces several of the mediators that lead to inflammation, some genetic predisposition, alterations in the mitochondria. So that means also in the energy function of the body, oxidative stress that is related to, as we have just seen, DNA damage and cellular senescence leads to cell destruction, cell loss, and inflammation. So all of that are the kind of are overall mechanisms that lead to inflammation and keep inflammation at a low grade throughout the individual's life. And that means that this inflammation, as it's kept, it's not resolved. So we do a cut on the skin, or we do a certain process, or a cosmetic process on the skin. We intend for this to cause a small acute inflammation that resolves and repairs, and we're going to see how this goes for the skin later. However, during our lives, the repeated processes of acute inflammation, as well as these low-grade inflammation processes we see here in the panel, they all lead us to have an inflammation, a long-lasting inflammation that leads to aging. Because all that we are seeing, the arrows going towards inflammaging, the inflammation per se, it also goes outwards and leads to more oxidation, more cellular loss, and so on and so forth. So everything that we see here related to inflammation, we are dealing with an inflammation that is kept at a low grade, but it's kept at a low grade due to acute inflammation happening repeatedly, but most importantly, it's kept at a low grade long term. And if it's kept at a low grade long term, it means that the all the processes that are related to inflammation, they keep happening. There's a cycle of them happening, and that leads to oxidative stress, that leads to function loss, and that leads to loss of the function of the cells, as we have seen in the loss of DNA as a whole. And this is kind of a good scheme for us to understand also and go back to the discussion we had last week in the previous session, which is inflammation is okay, inflammation should happen. However, when it happens in an uncontrolled manner, this leads us to a process that doesn't regenerate, doesn't solve. It might generate something that is not wanted. So we have a cut on the skin, and and by that cut on the skin, you can understand obviously, uh, um, any process we do to the skin. So any intentional damage we also might do in a cosmetic process to the skin as well might happen. It will elicit very similar mechanisms. So if that is well managed and inflammation is controlled, and if we have the correct ability to deal with this, what happens is that we have normal regeneration. So the normal regeneration would mean that the mechanisms that lead or the mechanisms that are activated and you see on the right side of the skin, that will lead to fibroblast proliferation and protein production. Namely, we have here collagen, but not necessarily only collagen. But what leads to the production of cells and tissue, this happens in a controlled manner. However, if we have more inflammation and we have more inflammation happening in an uncontrolled manner, as we have seen before in the inflammation process, this is this is localized also happening to skin, as we have, for instance, the scar tissue formation. So as if inflammation is not controlled, so if we have a stimulus to skin, or several stimuli, and this is not controlled, what happens is that we have scar tissue formation. This is very visible in the practice, but also I wanted you to see this as a longer term um, process in longevity and in the overall visibility or the visual visualization of how the body heals itself, so as to say. So that means, to summarize the hallmarks of aging and how it affects the practice, so as to say, it does look to me, and it does seem to be a consensus in the literature as well in the hallmarks of aging, that genomic instability is a center and also a beginning of the process of aging. But it also happens that all of that happens at the same time, and everything that happens later or after genomic instability might happen due to other causes as well, and also lead to genomic instability. And that is the reason why we will discuss this in our final discussion, which will be in the first week of August and also during the REMS event. Um, it is also the reason why the approach to longevity, or the approach to age or dealing with age, it can never be um, one-sided. It always has to be multimodal. It always has to be complete, because we are able to tackle some of these processes separately. Uh, and to be fair, anything we do in something that is so inherently bound to the human nature, everything we might do to prevent aging or to deal with longevity, all of that has, small effects in one process, a small effect in another process. Uh, and what we are aiming is to tackle many processes at the same time, so that we can reduce also this cycle that leads back to genomic instability, and obviously has a very important correlation to how patients age overall. And that is what I wanted you to have as a final slide for the hallmarks of aging, so that we can progress then to the other topics. So please, also, if you have any doubts, want me to come back to this slide, this is an important thing for you to have in mind. And then we go to the molecular aging. What do I mean by molecular aging? Is essentially the fact that we have some processes which we have just started to discuss both in the DNA and RNA class, but we have some very specific processes that happen during aging. So, as a matter of fact, we have discussed the central dogma of molecular biology, which is essentially the process by which DNA replicates itself, and it's related to cell proliferation, as we have started to talk today. It is transcribed into RNA, and in most situations, it is translated, or in many situations, it is translated to protein, and it has a function. So protein means function for us, but also RNA and DNA per se also have some functions in the cell. What is important from that? When we lose genomic instability, we are dealing with this, but also affecting transcription and affecting translation. So that means that during the process of aging, we will see damage to the DNA, but we can also measure how RNA and how protein quantities change across time, and what are the consequences of that. But that has begin with the DNA, with a very known part of the DNA, which is the telomere. So the telomere biology is something we discuss very often in the longevity sector. It is measured, it is discussed, but obviously it's not the only marker of aging, but it's a very important marker of aging conceptually and also practically. For those of you who haven't had a chance of looking at DNA per se, this is as fun as it gets. It's not very fun, but this is one chromosome and it's in its metaphysis part of the the cell division. So DNA overall is stained blue with dark here, and the proteins around the telomere they are stained red. So this is just a curiosity for you to visualize where the telomere is. But the most important part is when we have cell divisions, the tip of the chromosome is lost in most of the cells dividing. And why is this important? Because essentially, when we have the cell machinery we have discussed earlier, it is not able to replicate the tip of the DNA because the DNA polymerase 3 cannot bind to it efficiently. So what happens is that every time we have a cell division, unless the telomerase acts, which is not something we have in normal in all of our cells functioning. So unless the telomerase acts by adding some repeats to the end of the telomere, we lose a bit of the telomere. So we go on losing the telomere like that. So for most of the cells, once we reach a certain size of telomere, this means cells goes to senescence. What is senescence? These cells stop dividing. This is a normal process. But it also means that some cells will escape senescence, and that might lead these cells to either just be destroyed and generate inflammation. So first process, second process we discussed, all these cells might become a malignancy because we're losing here also telomere, also genomic stability. Why am I saying this in this situation? We have already seen how DNA is replicated, and this is one part of the DNA proliferation. But that also means that the telomeres per se, as they are in the end of the chromosome, they kind of keep the chromosome tightened as if we have a naggle on the as when we have a naggle on the tip of a shoelace. I think it's called naggle. Um, when we have this holding the tip of the shoelace, what happens is that it also prevents it from tearing and from separating and from breaking. What we have in a normal situation is that the telomeres are there to both protect the rest of the DNA and also to make us less prone to losing things that are in the middle of the DNA or the chromosome. However, any damage that happens to the telomere also might affect the rest or any stress damage that is not a physical damage or an oxidative stress damage that happens to the tip of the chromosome might also affect the rest of the chromosome. So as we lose telomere, we are more prone to different types of loss in the DNA. So what is important here? The telomeres that protect in the DNA, and it's a normal process that it goes on being lost, and we go on to cellular senescence, as in a normal process. However, if we break more the telomere, so we get subject to more breaking, more loss of telomere, faster loss of telomere, we might have too many cells going into senescence and also too many cells going into the escape senescence side, which leads to more damage to the organism and feeds back to genomic instability. Why is this important? Because this gets us as an idea, this gets us an idea of how the organism is aging. So the telomere here serves as a protection to the DNA, but also as a marker of how the DNA is being lost or is being damaged across time. And this has been shown in a very comprehensive paper in which we have seen that if we take enough samples, we will see that the telomere size always decreases with age, regardless of the type of cell. So for all the cells that keep dividing, we are always going to be losing telomeres, a natural process. What is in the natural process is what we are going to discuss later, is that as we add stressing factors to our cells, so as we add oxidative stress to our cells, as we add um, stress overall to the organism, we have seen telomeres to go faster. So telomeres are lost faster, and therefore that protective mechanism we have is lost faster. So this is the reason why we say, well, if you stress more the organism, if you're under more stress, and that might come from alcohol, that might come from inflammation, that might come from chronic stress, even in exercise, all of those things, they will lead to more telomere loss. Furthermore, it's also been shown therapeutically, and this is going to be the final aim for us when we finalize this molecular part here. Um, Providing things that protect the DNA or things that protect the organism from oxidative damage, namely antioxidants, they are able to decrease a bit how we lost telomere. So hold that information so that then we can go to the RNA side, which is the next step of that mechanism. So recovering a bit from our review, but also from the last encounter we have then, this is DNA, so the blue is DNA. And that means that obviously this is inside the chromosome, and also that means that this is what we might be losing if we are having loss of genomic stability. And this happens normally, but this is a process that is linked to aging. So we recall that RNA polymerase binds to the DNA and produces the RNA molecule, and then therefore produces protein. This is the function of the cell happening in real time. What is important? If we lose stability, we might lose a part of the DNA here, we might lose a part of the DNA here, we might lose the expression of the transcription factors, let's say. So many things can happen when we are in the process of aging that might lead to this thing here. So the simple thing here, as the RNA polymerase producing RNA not happening normally. And what happens is that as we age, we have seen some patterns that do correlate to the DNA loss, as we have the telomere, for instance, but also do correlate to how we lose or how we alter the RNA expression and the protein production. So this on the right side is what we have in several types of cells. We have seen different expression patterns. That means essentially RNA being produced differently as the mice aged. Obviously, this was done in mice so that we could see the lifespan going faster. But what strikes us as very important is that both in human disease samples and also in mice being accompanied throughout their lives, what happens is that first and foremost, we have many genes which are related to inflammatory response and control, and also to cell cycle as being changed across time as gene expression. So this is very interesting because obviously this emerges for us here as a consequence. Follow that very closely. So that emerges for us here as the consequence of DNA changing its behavior and leading to different RNA expression. But it also means that these are perceived in the research as things that are changing with the time, and they lead back to the function. So the tissue function also alters in response to that which we are losing or we are altering across time. So the important thing here to understand is that, well, gene expression changes across time. This is very well said. However, that goes from one side, which is the DNA damage leads, or the DNA stability or instability leads to alteration in gene expression. But this expression of the genes that were in some part of the DNA that was damaged, they will also lead to the aging process per se, because we are losing processes and functions in our body that might be related to controlling how our bodies age and how our cells adapt and maintain their functioning. In another study, it was also interesting to see that the aging signatures, they are also, if we take again mice here, as as a model here, we have also seen that inflammation again and the function of the immune system, they are very much well related to how aging happens. So that means that once our body starts not being able to control inflammation well, because we are losing expression or altering expression of many of those genes, that leads us back to the aging again. So this is another takeaway message from this part. And finally, here we had this other study, this last study I wanted to present you, in which aging, um, the important thing here is that when we take different parts of the age of this model organisms, it was possible to see that inflammation and senescence associated genes, they became they become progressively different across time. It's not like it's something that happens once, but it's something that as we lose the capacity of controlling the expression of these, essentially inflammation and cell proliferation, these are things that we have seen in the beginning as reasons for us to age, but also that means that when we lose the capacity of doing it, that also leads us back to the aging process. So this is the last message I wanted you to have here with respect to RNA expression and aging. So that all means that the molecular aging, which was the title I gave to this part of the conversation, so the molecular aging per se happens, and it might happen either as a response to DNA damage, or it might happen leading to DNA damage. So this is something that might be happening in a cycle, everything. So molecular aging will mean that we have telomeres shortening, we have epigenetic alterations and transcription alterations happening, that leads to cells not functioning the same way. So the tissues will not function the same way, will not recover the same way. And finally, we have age-related disease, so which are, overall uh, um, visualizations and clinical perspectives of aging, and also the aging process as a whole. And that takes us to the regenerative capacity, which leads us to finally discussing for us to close what can we do. So we have spoken so far about the mechanisms. We will have a dedicated session in the end, which would be our last session, really to the regenerative mechanisms and their molecular correlations. But here I'd like to advance to you what is related to telomere, what is related to RNA expression. So how to promote this regeneration, what happens during regeneration. So the first thing for us to cover here is that we do have telomere loss to one side, and this is mainly going to be both related and fueled by oxidative stress and inflammation. So oxidative stress leads to telomere loss. Chronic inflammation leads to oxidative stress and telomere loss. What can happen here? What can happen here is that we do have some ways of protecting the DNA. So oxidative stress is counteracted by the presence of antioxidants, and therefore this is a therapeutic aim and focus here. But not only this, as well as we have obviously the control of inflammation as another possible focus for us to counteract aging. So I'm not saying here that we're going to give anti-inflammatory to patients because that would trigger other alterations. What I'm saying, and what I want you to remember is that when we have the concept of inflammation and not inflammation, what happens is not that we have a burst of inflammation and we control it, is that we have a long-lasting inflammation that leads to both stress in the organism and also loss of function in that organism. So what we are talking about here is to keep the organism controlled in terms of the quantity, um, and I'm talking about real amount of antioxidation capable of counteracting directly oxidation, and also anti-inflammatory agents that will be able to counteract this sustained period of inflammation which we have, and obviously that should be controlled so that we control aging per se. Um, as we age, one of the sides of it is the telomere, and the other side of it is the overall cellular function, as we have discussed. And we have many factors happening, or many processes happening in our cells that also lead to aging, and also can be counteracted by normal measures and also focused measures, so as to say. So there's one of the consequences of when we start losing cell function, is that we have alterations in the mitochondrial function, so the mitochondrial dysfunction, which arises from which arises and it's correlated very closely to essentially the um, the energy metabolism as a whole. And on the other side, what we have is also the fact that as cells express genes differently, the sensing or the reception, the mechanisms of signaling coming from outside the cells related to the nutrients also become different, and they uh, um, also become a challenge for the cell. So what happens here is the following. So these are two mechanisms that are very intrinsically linked to the cellular function. And obviously, there's not much we can do to mitochondrial function in general or very focused, because we cannot go and just go on and alter the function of one mitochondria of one cell specifically. And the same goes to the fact that once we start losing cell function and we start having cells not sensing nutrients very well, that means um, that essentially we need to tackle these cell functions altogether. And what I mean by that um, is that one of the possibilities, or one of probably the clearest possibilities we have seen functioning here, is that as simple as it sounds, calorie restrictions and ketogenic diets, they are able to both alter the mitochondrial function and also the sensing of the cells, or at least to counteract the loss of cell function we have. So we have seen these as two very important mechanisms of keeping patients and keeping patients from aging as being very important ones. So those are kind of lifestyle alterations that in the end are very important to the overall function of the cell. So decrease calories and ketogenic diet can be employed therapeutically in the concept of longevity. These are very important themes here. We have newer targeted antioxidants and functions, such as the NAD plus and NAD way of dealing with the mitochondrial function. So this is another focus of therapy. So this is another focus of therapy that has been very importantly related to longevity. And finally, we have the mTOR inhibitors, which we're going to discuss in our last class. This is controversial, but this is also very well established as having also anti-aging effects. So we are losing cell function, we're losing DNA, we're losing cell function, we might be losing the capacity of cells to sense nutrients and to produce energy efficiently, and that takes us to solutions. And the main solutions here are calories restriction, keeping the antioxidants and keeping the mitochondrial function with NAD and the targeted antioxidation. There are other options here. There are other research going on, such as the alkanase inhibition, but these are less frequently and less well proven yet in the literature. That takes us now for us to finalize this theme. That takes us now to kind of the visible way of dealing with regeneration. So the regenerative capacity of the skin is something that is very well desired. So we have seen what happens in the cells overall the body, and we have spoken a bit earlier on how the skin itself regenerates. And probably the most important thing here is that the wound healing process is always going to be present whenever we do something to the skin of a patient, aiming at improving the skin. So what we want to happen is that cells must proliferate and reestablish the tissue. However, we do have some underlying processes which are essentially local inflammation processes that will lead, if not controlled, so if the inflammatory phase is not controlled during skin proliferation or during skin regeneration, what happens is essentially that we have tissue loss or tissue function loss. And that is very much linked to the type of immunity that's happening and the type of cells that reach the skin at that time. And essentially, what we have also seen is that as we age, the capacity and how the cells proliferate in that tissue, they are different. So we have seen previously that we lose the capacity to control, or we lose the capacity of expressing some of the proteins associated to inflammation and to the immune system control. What happens is that when we age, also the capacity of regenerating the skin is visibly different, and this is something that obviously is going to encounter you in the clinical practice. So some things happening in the skin, which are a reflex of how much this chronic inflammation and this loss of function and this alteration in gene expression in the inflammatory system, how that really impacts the patients being treated. So, as you know, the capacity of skin regeneration will alter as individuals age. And part of that is really due to the fact that the signaling or the inflammatory signaling and the control by the immune system changes across time, as we have seen in the gene expression profile. So we become less able to control inflammation. That means that apart from that, or that means that due to that inflammation, as patients age, the efforts on the skin to make it regenerate out of making an inflammatory process. So that means that when you are doing a process to an aged skin, so as patients age, um, it is desirable that you promote less inflammation to that patient. So that means go milder on these aged patients or aging patients, so that the skin is able to heal better. So this is the reason why this is a logic in the regenerative cosmetic aspects of skin regeneration. It's really because we become less able to recover the skin as we age. And this is a very important aspect in the clinical practice. So as a conclusion here, both from this part, but also the utility of using regenerative medicine as wounds age and as individuals age. Oh, sorry, that was repeated. Let me just grab the correct conclusions here, just so you can kind of screenshot the final decision here. As we age, then we have seen alterations in how individuals control inflammation, and that is going to be a very important aspect um, of how essentially um, how essentially um, how essentially we recover both the overall function of the organism, but most importantly here on the skin, because this is something very visual for us to see happening, or something very visual for us to also measure in the patient's satisfaction out is. However, obviously, as we have seen previously, all these systems, and that means essentially um, all the systems, and that means essentially coming from DNA damage to RNA expression and so on and so forth. So as we age, we also are less or more or less able to control um, how um, how this regeneration happens. And this means that telomere attrition or telomere loss, epigenetic alterations, transcription alterations, they are going to govern how individuals are going to age. But it also means that regeneration depends on we being able to control inflammation, control regeneration, and control the overall loss of DNA. So controlling this from different points is probably going to be ever the best solution ever for the longevity of the patients. Well, it was a bit longer than expected, so I'm going to take my presentation out just so I can see if any of you have questions, and we can actually discuss a bit um, if you have time.
Thank you so much, Gustavo. That was an excellent presentation, as always. Does anyone have any questions? If you do, pop them in the Q&A box now and we can get some answered. Um, doesn't look like we've got any coming in, but I would, would welcome if you do have any thoughts after the presentation. If you want to email us, info@raminstitute.com, then feel free and we can pass the questions on to Gustavo. Obviously, he'll be back here next week for our fourth in our genetics and regeneration series. Do join us then. Same time as always, 7 p.m. UK time. Yeah, we don't have any questions, Gustavo. Is there anything you'd like to add? Any sort of key messages you'd like to give after tonight's
Um,
presentation?
yes, we have. I'll have
Or
here
just
finally
one
a
has just
question.
popped
So,
in?
introduction of exogenous antioxidants can unbalance the endogenous antioxidants activity. Um, I wouldn't say this. Yeah, there is a possibility always, in exaggeration or in, enormous quantities of antioxidants that can unbalance the antioxidant activity, and also has been shown in some infections, for instance, to unbalance how the immune response happens. However, in normal quantities, in normal therapeutic quantities, in the antioxidants we use in the clinical practice, I wouldn't say this is something to be worried about, especially because in nowadays lives, we are always kind of introducing stressing factors, and these stressing factors, they are very much connected to oxidation. And one example I always talk about in real practice, both with patients and other practitioners, is the fact that sometimes the damages we inflict during regenerative processes, as well as very importantly, um, excessive exercise, they all lead to a huge amount of oxidation happening in the body. And therefore, I don't think, and this is really me thinking here, because there is no clear evidence from that side, but I don't think there's an amount of antioxidants that is used in the clinical practice that would unbalance, importantly, the endogenous function of oxidation.
There we are. Hopefully that has cleared that up. If there's nothing else, then we will leave it there for this evening.
Thank you very
And
much, Wilson.
we will join you all again next Wednesday, 7 p.m. Like I said, if you've got any questions, whiz them over to us, info@raminstitute.com. This webinar will be available on our website, and we'll send the link out in the next few days. So if you did want to rewatch anything back to remind you of anything discussed, then please do so. Thank you, Gustavo, and thank you everyone for joining
Thank you all
us.
very much. Thank you all very much. I'll see you next week.
See you next week. Thank you. Bye.
Bye, guys.